Thanks a lot, Adriano. I hope you can hear me, guys.
Absolutely, perfectly well.
Okay, great. So thanks a lot for the invitation. I hope everyone is doing well despite the pandemic. And I'm really happy today to give you some insights into how we actually in discovery pharmacology use humanized mouse models to profile our novel antibody drug candidates for the development in the area of cancer immunotherapy. So we have heard that there are some different definitions, right, of humanized mice. So I just have one high-level introduction slide on how we actually produce our cohorts of humanized mice, and also why we think that these mice are a pretty good tool to especially look into T-cell targeting cancer immunotherapies. So on the left-hand side, in this cartoon, you basically see the robust and very straightforward protocol of how we humanize juvenile energy mice. We treat them with busulfan to actually clear the stem cell niches, and then we inject CD34 positive human hematopoietic stem cells. And then when I'm talking about humanized mice today, this is what we actually get after around 3 to 4 months, which is a mouse that actually carries a human-like immune system. And some high-level characterization is shown below. So when we screen those mice in blood, for example, you see pretty high engraftment rates, so high numbers of CD45 positive human immune cells, and also nice differentiation into different lineages. So you see CD19 positive B cells, and also, and this is for us of major interest, CD3 positive T cells. And beyond that, what is really also quite remarkable is on how well these T cells in these mice actually differentiate. So you see CD4, CD8 cytotoxic T cells. Also, if you look at the lower panel in spleen, for example, you can also detect alpha, beta, gamma, delta T cells and regulatory T cells, for example. And what's quite remarkable, imagine this, these are human T cells in a mouse, right? Nevertheless, they exhibit a pretty naive phenotype, as you can see by lack of granzyme B and Ki67 expression. And the reason for that, and one of the most important features of the stem cell humanized mice, is shown on the right. So at the end, the T cells, they get positive, negative selected on the mouse epithelial tissues. So you can see some nice characterizations here done previously, where you can see in the thymus of these mice the double positive thymocytes, which actually come from the bone marrow. And then you can see a nice cortical and even medullary structures, similar to humans, by the way, where actually the thymocytes get positively and negatively selected. So the outcome of all of this is quite similar to what you have in humans, which is that the T cells aggressing from the thymus are basically tolerant to the host, which is, of course, in this case, a mouse. So why is this so important actually for us? You see a kind of a characterization of the T cells on the bottom here. So in blue, you have the T cells isolated from humanized mice, and on the top in red, you have the T cells isolated from human blood. And as you can appreciate, the pattern looks quite similar. So if you look at different activation markers like PD1, 4-1BB, or also CD69 at baseline, and this you can see in black, those mice exhibit a naive phenotype similar to the T cells in human blood. And when you now, for example, want to test some T cell bispecific antibodies, for example, which start to engage the T cells and activate them, you can likely do this also with T cells in a humanized mouse, as you can see here in the colored histogram. So when you add a CD3 targeting antibody, the activation markers are nicely upregulated, and the pattern is quite comparable to the pattern that you would see in a human being. So the humanized mice generated by stem cell transfer, in our eyes, are a pretty good tool model to validate, for example, T cell engagers. And I just wanted to tell you kind of a little story about one of those molecules that we have actually also already brought to the clinic. So that molecule is in phase 3 clinical trial at the moment, and humanized mice were quite essential to profile this molecule in preclinical settings. So the molecule I'm talking about is glofitamab, which is a CD20 T cell bispecific. So you can see on the left-hand side that molecule has 2 binding arms for CD20, which is highly overexpressed, for example, on lymphoma cells. and a CD3 binding arm that actually binds to the T cells. It also has a silenced Fc part to just render those molecules less susceptible of binding to Fc receptors and prolong the half-life. And what's quite nice about this format is the mode of action is pretty potent. So on the right-hand side, you see a nice cartoon where you can see that if you have a CD20 positive tumor cell and a CD3 positive T cell, and you add the CD20 TCB to the system, you actually cross-link the T cells to the tumor cells, which then start to secrete cytotoxic granules and finally start to kill the tumor cells. So at that time, we were basically asked if we can show an in vivo proof of concept for a human lead molecule binding to CD20 and CD3. So we said, well, the humanized mice, stem cell humanized mice might be something because they have a lot of endogenous B cells and also endogenous human T cells. So in a very first study at that time, we tried to just cover basic parameters of this molecule in vivo. So we used humanized NSG mice. We injected clofitamab IV into these animals and took blood after different time points to basically look at the PK and also the PD of this molecule. So in the middle graph, you see the exposure in mice, and if you focus on the circles here, these are the humanized mice, and you can appreciate that clofitamab has a pretty favorable PK profile, a very long half-life. Interestingly enough, also if you compare the PK profile to normal NSG mice, where you don't have CD20 and also you don't have CD3 present, right? Because you lack the endogenous immune cells, the PK profile looks quite different. So the target-mediated drug disposition effect could here nicely be reflected only in humanized mice. And when we then looked into blood and looked for the PD effects of this molecule, we already saw quite some striking activity. So you can see that the endogenous B cell counts, they drop significantly upon injection of this bispecific, and also the T cells. And this is known that if you activate T cells in circulation using a CD3 binder, they start to transiently marginate, right? So they leave the bloodstream and come back after a certain while. So these were very first encouraging results to actually show that indeed it seems that the CD20 TCB can cross-link CD20 positive target cells and CD3 positive T cells. So in the next step, of course, we wanted to dig also a little bit deeper into understanding the mode of action. And on the left-hand side, you see that 72 hours after the infusion of this antibody into humanized mice, you see really a nice activation of different T cell subsets. You can see upregulation of PD-1, Ki67, which indicates that T cells start to also massively proliferate. And also nice, of course, when you think about an anti-tumoral response is the upregulation of granzyme B in the CD8 cytotoxic T cell compartment. So the next step, of course, we said at the end, this bispecific should be able to recruit T cells to a tumor and eliminate the tumor cells. So this is why we then inoculated a lymphoma cell line called WSU DLCL2 subcutaneously in humanized NSG mice. And you can see in blue, these tumors grow very aggressively. And we then tried different conditions where we basically added CD20 TCB at different doses, and you can see a nice dose response. And already at that time point, we figured out, okay, that molecule seems to be quite potent because even at intermediate doses, at the end of the day, all tumors were forced into regression. So and then there's another aspect of the story, which is also interesting to tell you. So when we started treatment in non-tumor bearing mice, we realized that mice really all of a sudden lost a lot of body weight within the first 24 hours after treatment. They finally also recovered, but it was quite tremendous. And of course, we tried to understand what's happening. And on the right-hand side, so you basically see the explanation, and this is known also for T cell engagers. The engagement or the cross-linking of the T cells to the CD20 positive B cells leads to a massive cytokine storm in these animals, which is very pronounced, especially at 24 hours after the treatment, but then afterwards declines, which is also, I think, nicely reflecting the body weight drop kinetics. So at that point, we already said, well, I mean, that might be also an issue in patients, right? Because also patients have a lot of CD20 positive endogenous B cells, and of course, CD3 positive T cells. So we said, okay, let's already think early on about a possible mitigation strategy to that. So one idea we had is, okay, maybe if we debulk before we actually infuse the T cell bispecific, the endogenous B cells, you might get rid of the cytokine release syndrome. So these data are shown here. We compared, on the one hand, humanized NSG mice without a tumor at that time upon infusion of glofitamab, and you can see again this really huge cytokine increase, especially at very early time points. And then on the right-hand side, you see the same kind of picture, but with a pre-treatment using an anti-CD20 antibody, which is called Gazyva. And this antibody basically depletes all the CD20 positive endogenous B cells. And as you can nicely appreciate on the right-hand side, the cytokine storm was completely abrogated upon debulking of the B cells. And I think this is a very nice example because, of course, people often ask, okay, it's nice that you actually generate mouse data, but has this any clinical implication? And when this molecule entered clinical trials, the first thing people observed, even at very low doses, was serious CRS. So and then they went back and also based on this data here we generated in humanized mice, they also explored Gazyva pre-treatment in patients. And still now in phase 3 clinical trials, Gazyva is being applied 7 days before the infusion of glofitamab in refractory lymphoma patients. So just saying that these data here were really highly translatable and also really relevant to actually mitigate CRS in cancer patients. Then another important question that you usually get is, okay, can we even boost the efficacy of such a molecule? Because a T cell bispecific binds to CD3 and provides a nice signal 1 to the T cells, right? But if you follow the T cell biology, you know that you also at some point need a co-stimulatory signal, so a signal 2, to actually keep them activated. And one of the most powerful co-stimulatory pathways for T cells is the 4-1BB and 4-1BB ligand pathway. So the people at Roche, they designed a molecule which actually binds to CD19, also on the lymphoma cells, and on the other arm of the bispecific antibody carries the 4-1BB ligand. So the idea is that this antibody can finally bind in the tumor context to activate the T cells, which start to upregulate 4-1BB and just sustains the activation status. We then did another in vivo efficacy study, as you can see in the middle here, the waterfall plot, where we treated tumor-bearing mice, also again lymphoma, CD20 positive lymphoma, xenografts with CD20 TCB in red, with CD20 TCB plus a non-targeted 4-1BB ligand and also a CD19 4-1BB ligand molecule. And as you can see, again, CD20 TCB was quite good in using some tumor growth inhibition. Nevertheless, if you look at the purple graph here, I mean, the combination with the co-stimulatory molecule was able to actually drive most of those tumors into deep regressions. It's also quite remarkable if you look at the immunohistochemistry of the tumors in these studies. So you can see in the middle panel the human CD3 staining, which actually detects the human T cells. And as you can see nicely, that glofitamab alone was already quite good in attracting some T cells, but if you follow and go to the very lower row, you actually see in the middle that the combination with CD19 4-1BBL actually leads to a exponential expansion of the T cells on the tumor site, which is then also correlated with a much lower number of CD19 positive tumor cells. Based also on these very encouraging data, the team finally also decided to test this combination in the clinics. And we have a current phase 1B study ongoing where actually glofitamab is combined with CD19 4-1BBL in refractory lymphoma patients. Another thing that comes to your mind when you think about a bispecific antibody is relapse. Why? The main reason for this is that, of course, not all the lymphoma patients across the board have very high and homogeneous CD20 expression, right? So we tried also to address in humanized mice very early on this possible relapse that you might also experience in the clinics by using a cell line that is called OCI LYY18 that has a very patchy CD20 expression. And on the left-hand side in black, you can see that the glofitamab treatment actually resulted in a pretty good initial tumor response, right? So there were even regressions, But finally, most of those tumors grew back, most likely because some of those tumor cells just did not express the target CD20. When we added at the time point close before the relapse, the CD19 4-1BB molecule to actually support the initial T-cell response against this lymphoma xenograft, we could nicely show that this indeed also helps against pretty hard to treat lymphomas in a way that it can keep really the relapse under control. And on the right-hand side, you also see that finally you end up having a statistically significant prolonged survival. So this was the story of glofitamab. I think this is our most advanced molecule, so to say, that we also have tested in humanized mice, and where we did a lot from a set initial PK until really these kind of clinically relevant relapse settings. To also show you two additional examples, I would also like to highlight that we use humanized mouse models also to really evaluate proposed mode of actions of our next generation CIT molecules. So the first example I would like to show you is a T-cell bispecific again, but we call this next generation T-cell bispecific. And you might ask, okay, why next generation? What was wrong with the first generation? Actually, it was quite potent, but this is also a big issue because, I mean, for a very potent T-cell engager, you really need a very, very clean target. So the target ideally is only expressed in tumor cells, but not in healthy tissue, because you end up having on target off tumor toxicity, which certainly at some point also will result in dose limiting toxicities. So then the team at Roche came up with a pretty interesting approach. So they said, okay, let's try to design an antibody where the CD3 arm is only released when the T-cell bispecific is bound to tumor cells, so in the tumor microenvironment. And the way of how they did it is shown in this cartoon. So the antibody targets folate receptor, which is also highly expressed, overexpressed in cancer, with two binding arms again. And it also has a CD3 arm on top, right? But this CD3 arm is masked. You can see this. There's another antibody, an anti-ideotypic antibody binding to the CD3 arm and basically blocks the engagement of the T cells. So the clue about the system is that it has a cleavable linker, so this anti-idiotypic CD3 antibody. And whenever there's enough metalloproteases present, that linker is cleaved and the CD3 arm is released, which then finally can bind, again, the CD3 positive T cells. So the idea basically is that in normal tissue, where you might even have folate receptor expression, because of the lack of high density of metalloproteases, the antibody stays like it is on the left-hand side and is not able to recruit T cells. But when it comes to the tumor and it binds to the target on tumor cells, there's enough protease activity to actually cleave that linker and to make the T cells also then accessible to the tumor site. At that time, we struggled a little bit, to be honest, to find an appropriate in vivo model, just because we realized that most of our cell line-based models, they actually did not express protease at all. We then established different patient-derived xenograft models, and here shown as an example of a breast cancer patient-derived xenograft model, where we realized that the protease baseline expression is much higher, and also we were lucky enough to identify models which on the other end also expressed the target, which is the folate receptor one. We then designed a first proof of concept in vivo study. The cartoon is shown here, where we engrafted the breast cancer PDX model autotopically into humanized NSG mice, so in the fat pad. And then we performed an efficacy study where we checked for the effect of different molecules. So on the one hand, we checked, of course, for the molecule that has the cleavable linker. So in here we had two different formats called MAT-C and MAT-B, which are basically two different cleavage sites for the proteases. Then we had the parental folate receptor TCB with the free CD3 binder, and we had also a non-cleavable version, which means that this molecule also has the mask to capture CD3, but also in presence of high levels of proteases, that CD3 binder is not released. And then on the right-hand side, the very first thing, of course, we had to check is whether especially these masked molecules, they are stable in circulation and they don't get unspecifically cleaved. So they did some bioavailability assessment in serum of the humanized mice with and without tumor. And what you can nicely see is that only the molecule that has a free CD3 binder, which is the parental one in red, the folate receptor TCB, can be detected. So a free CD3 binding arm can be detected in circulation, whereas the two masked molecules actually did not show any free fraction of CD3 in the circulation. So, and of course, in the next step, we also had to check for efficacy, and we treated those patient-derived breast cancer bearing mice with the parental TCB, which is shown in red, which usually induces quite nice tumor growth inhibition. And you can see that even the cleavable TCB with the MAT2B format, it's giving you an equally good efficacy, whereas the other format, the MAT2C, actually has quite some effect, but it's far below the efficacy that you gain with the parental T-cell bispecific. And of course, importantly, also that the format where basically the mask is not cleavable, you don't see a lot of efficacy as expected. On the right-hand side, we've also confirmed this kind of a trend in TGI by looking at intratumoral PD. So where we can see that as compared to the vehicle in black, the parental TCB, as well as also the MAT-B format of the protease activated TCB, gives you a very nice infiltration of human T cells into these tumors. So a very, I think, early on concept. So let's see if these molecules can finally improve also the safety, especially of the T-cell bispecifics in the clinics. But at least in this mouse model, it really looks like you can selectively activate a T-cell bispecific in the tumor context, but not in the periphery. And then last but not least, because you might now say, okay, humanized mice work quite nicely for T-cell bispecifics, but can we do anything else? And the answer is yes. So we have in-licensed a very interesting molecule, which is a Treg depleter, so a molecule that aims to deplete regulatory T cells. Of course, ideally, especially in the tumor context. And the molecule is shown on the left-hand side, so it's not as fancy as the TCB, the next generation TCB. It's a simple IgG format where you have two binding sites for CD25, which is the IL-2 receptor, which is known to be very, very highly expressed on regulatory T cells. But it blocks not the IL-2 signaling, and that's very special about this molecule. It binds to the receptor, but the IL-2 signaling still is possible. It has also a modified Fc part to enhance actually the ADCC and ADCP in the tumor context. So then on the right-hand side, there were two things that the team proposed, right? On the one hand, this antibody, by binding to CD25 in the tumor context, should deplete the Tregs, right? And then by depletion of the Tregs, which are the main consumers of IL-2 in the tumor microenvironment, the idea was, okay, you release really a lot of IL-2, which then becomes accessible to the CD8 positive T cells, which also express very low levels of CD25, but also a very low affinity variant of the receptor. And usually, this is not enough to actually compete with the Tregs with these high CD25 levels in the tumor microenvironment. So we had these two questions. Can you show us that this molecule indeed depletes human regulatory T cells? And on the other hand, can we show that this depletion actually has an effect on the cytotoxic T cells? And at that time, I mean, we felt this was a really big challenge because Treg numbers in humanized mice, especially in tumors at baseline, are really, really low. So, but nevertheless, we screened a lot of different tumor models, and we realized that the infiltration of Tregs into tumors is very much dependent on the tumor itself. So we realized that a pancreatic tumor model called BXPC3 at baseline already has quite some high numbers of Tregs. So we were pretty happy because we said at least we can look at these target cells. And then we engrafted these tumors subcutaneously into NSG mice and injected the Treg depleter, so the anti-CD25 antibody at different doses. And we took down scouts at different time points and checked for Treg depletion and also CD8 phenotype in blood, spleen, and also in the tumor. And a representative picture is shown on the right-hand side. So on the one hand, you see a very great effect of the Treg depleter when it comes to the frequency of FOXP3 CD4 positive cells. So they are more or less gone upon the injection in tumors. And also what you can already see here quite nicely from the density plot is that the CD8 positive T cells actually start to upregulate quite significantly granzyme B, which leads finally, of course, to improve tumor cell killing if you think about tumor context. So if you look then finally, and this is my last data slide, don't worry. If you look at this in a longitudinal fashion, so on the top you see the Treg frequencies in blood, spleen, and tumor. And on the bottom, you see the frequency of granzyme B in CD8, so activation of T cells. You can nicely see that you deplete, indeed, the Tregs in all organs tested, and also you do this in a dose-dependent fashion. And similar to the dose dependency when it comes to the Treg depletion, the granzyme B expression in CD8 positive T cells go up. So we were quite happy because we really were not that sure about the role also of Tregs in humanized mice and about their functionality, but it really seems that you can also, in certain settings, study even this very rare subset of T cells, which have, of course, also a very important function in tumor biology. And I mean, these data were also quite helpful, by the way, for the clinical team at the end, because they used the PK-PD relationship seen here in this study for the entry into the human dose prediction. And also what the team is doing at the moment, because that molecule is phase 1 dose escalation at the moment, the key biomarker at the moment they are looking at is the depletion of the Tregs in blood, because also here we saw nicely that this is a very good correlation between the Treg frequency in blood, the Treg frequency in tumors, but also the mode of action, right, of this molecule as seen by activation of CD8 positive T cells. So, and this brings me already to my last slide. I hope I was in time, and I hope also I could show you a little bit on why we think actually that stem cell humanized mice are really great in vivo to profile cancer immunotherapy drug candidates. You can use your human lead molecule, which is of course a big advantage over syngeneic models. And also you can study this molecule in the context of a human tumor environment by using cancer cell lines or even better PDX models. And there are also, one has to say, fundamental differences in mouse versus human biology. We have, for example, also tested a mouse surrogate molecule for glofitamab, so the CD20 TCB, but it was actually very hard in a syngeneic setting to reflect what at the end was happening in humans. Of course, you can assess also human relevant biomarkers as shown by for example, the cytokine release for glofitamab. And overall, as said, and this is also highlighted by the picture on the right, you really think that the humanized mice, if you ask the right question and you understand the mode of action of your molecule, can really bridge between normal animal models, so syngeneic mouse models, and the cancer patient.